Steps of the translation process. Initiation. The small ribosomal subunit binds to the initiator tRNA carrying the initiator amino acid methionine. This complex then attaches to the cap structure at the 5' end of an mRNA and scans for the start codon AUG. The process is mediated by several initiation factors. At the start codon, the large ribosomal subunit joins the complex and all initiation factors are released. The ribosome has 3 sites. The A site is the entry site for new tRNA charged with amino acid or aminoacyl tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anti-codon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right aminoacyl tRNA enters the A site, a peptide bond is made between the 2 now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves 1 triplet forward on the mRNA. As a result, the empty tRNA is now in the E site and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new tRNA. The cycle is repeated for each codon on the mRNA. Termination. Termination happens when one of the 3 stop codons is positioned in the A site. No tRNA can fit in the A site at that point as there are no tRNA that match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is dissociated into subunits and is ready for a new round of translation.